All right, we've been looking at how genes lead to proteins and physical traits, but that's not the whole story. Your genes can actually be regulated on when they are expressed, when certain proteins are made, when they're made more frequently or more in greater quantities, and when they're turned off completely. Let's look at how that works within our body, and we're going to start on a very simple we're going to start with a very simple model in prokaryotic regulation. Remember, prokaryotes, they do not have a nucleus. They have circular DNA, and it's much simpler. So let's start there. We're going to look at something called an operon. Operon. This is a group of structural and regulatory genes that function as a single unit. And here are those pieces to an operon. Each one of these is part of an operon. You have the promoter. This is where RNA polymerase, remember we talked about polymerase was what builds an RNA molecule. The promoter is a part of the DNA sequence, so it's on your DNA, where RNA polymerase is going to attach. And it's just a small segment, which I'll show you on the next slide. And then you have the operator. This is going to be a small piece of DNA where a repressor can bind. And just like the word sounds, a repressor is going to repress or turn off a gene. And this is just a short segment of DNA as well. I'll show you on the next slide. Structural genes come next, and that's going to be one to several genes. It could be many genes coding for enzymes on a metabolic pathway, which we've talked about in a previous chapter. And those are transcribed simultaneously as an entire long segment. And then you have the regulatory gene. That's going to code, it's going to be the DNA code to make repressor proteins. So the code to make the repressor to turn off this whole operon is found within the operon, which is kind of crazy to think about. The regulatory gene is normally located outside of the operon, however, but it is included in the whole process. Okay, and here's a visual so we can look at. So this is going to be prokaryotic regulation. This entire piece is an operon. We have the regulator gene, which is going to produce the repressor. Okay, we have the promoter in a different part. So here's the entire operon right here, this whole segment that's together. The regulator gene in this case is not within the operon. However, what it does, the important part, the regulator gene, is going to be the code to make this protein. And this protein is a repressor. When this repressor binds to the operator, don't get operator and operon mixed up, it's easy to do. The operator is actually this little segment of DNA right here in purple. And when the repressor binds here, then the RNA polymerase, which binds to the promoter right next to it, is not able to bind to the structural genes to produce this mRNA. It's not able to transcribe the structural genes. So I'll show you that here. Right here we show that the repressor is bound to the operator region in this section, and it's blocking this RNA polymerase from binding. And RNA polymerase is going to be the enzyme that transcribes the structural gene here. So it's going to be responsible for making all of these enzymes in a metabolic pathway. In this case, what we're looking at is tryptophan. So these enzymes are needed to synthesize tryptophan. Tryptophan is produced by a metabolic pathway requiring each one of these enzymes. When tryptophan is present, then it binds to this repressor, and the repressor then binds to the operator, turning the whole process off. So once there's enough tryptophan, then this gene is actually turned off by the repressor. It's kind of cool. Self-regulating process. I'll show you another example down here. This one's going to be more familiar because it has to do with lactose. So lactose is going to be an enzyme that breaks down, okay. Lactase is the enzyme. Sorry, I'm reading that wrong. Lactase is going to be an enzyme that breaks down lactose. So this right here is going to be the regulator gene, that would make the lactase enzyme, or the lactose metabolizing genes. That makes lactase enzyme. So when lactose is not present, so there is no lactose, then we don't need to make lactase. We don't need to make something to break it down if it's not even there. So there's no point in wasting energy on producing this protein if it's not needed. Therefore, as you can see, the repressor is bound to the operator and this gene is turned off. RNA polymerase cannot bind to the promoter, it's blocked. Okay, but down here, let's say you just ate ice cream. Now you have lactose in your system. Lactose will actually bind here to this repressor gene, to this repressor protein, and that protein will change shape and fall off of the operator. Now there's nothing blocking this RNA polymerase from binding to the promoter region, and voila, here's the mRNA polymerase, and this lactase gene is actually transcribed, and lactase is formed so that it can break down the lactose. Now, as we know, some people do have lactose intolerance, which means they're not able to make enough of the lactase gene. So there is something has broken down in that pathway. Okay, so that what we were talking about was prokaryotic regulation. Let's look now at eukaryotic regulation. Now, eukaryotes are more complicated. There's a nucleus involved, and we go through transcription and translation, as we have been talking about in previous chapters. So there's a variety of different mechanisms that can regulate genes within eukaryotes. What you guys need to know are these 5 primary levels of control. And if we kind of break it down into where these levels of control take place, it'll help you keep them separate. The first one is to look at what happens within the nucleus, the nuclear levels. How are we able to turn genes on and off, or these proteins really, on and off, make them not be expressed or be expressed within the nucleus, so before they even leave the nuclear membrane, we can turn these genes off. The first one is going to be chromatin structure. Remember, chromatin is DNA in its active form, where it looks like a bowl of spaghetti we've been talking about. Pieces will be wrapped around histones right here, and some pieces are left free in order to be transcribed and translated later. And then there's transcriptional control, and this is going to be RNA polymerase binding, like we just talked about before. Is RNA polymerase able to transcribe that piece of gene? And then there's post-transcriptional control. Once that RNA polymerase has made the pre-mRNA, is it going to be turned into a mature mRNA that is able to leave the nuclear membrane? That's where post-transcriptional control takes place. And you can tell from the name, post meaning after transcription. So this is a control during transcription, and this is a control after transcription. But it's all still within the nucleus. And then you have the cytoplasmic level. So once it leaves the nucleus, and now the process is out here in the cytoplasm. What happens in the cytoplasm? Translation happens in the cytoplasm. Because translation happens on those ribosomes, remember? And ribosomes are going to be located out in the cytoplasm, outside of the nucleus. So we'll see some translational control would be, is the mRNA going to be translated into a protein? Or is it going to be broken down before it is translated into a protein? And then you have post-translational control. So once you've got, remember it's not a protein yet, it's a, yeah, I can't think of my words. Where was I going? It's a long chain of amino acids called a polypeptide chain. I don't know where it is, right here, I guess. Here it is. It's a polypeptide chain. Until it's folded up, it's not a protein. So you can still stop it there. And once it's a protein, you can have it break down immediately. Why is it important to be able to stop the process at each one of these levels? Because we need to sometimes be able to hit a kill switch to stop it. we don't need it anymore. We want it to stop immediately and quit wasting our resources. So there are processes where you can stop the translation or the transcription of certain genes at each one of these levels. I'm going to look at some of them in more detail. You have chromatin structure. Remember how if we have our chromatin wrapped really, really, really tightly around these histones, then we are able to pack it tightly enough that it will not be transcribed. Whereas you have some pieces that are left out, and those are open for RNA polymerase to bind to, and they can be transcribed that way. Nucleosomes are these wound around groups of 8 molecules of histones. So they're just groups of histones, and they're tightly, tightly wound. These will not be transcribed. It's a way to turn off those genes on that piece of DNA. Okay, so it all has to do with which genes are accessible. Are they even accessible? Are they out, or are they packed away? You can also look, and this is some names that I've thrown out here before. You guys have seen these before. But euchromatin, these are going to be the genes, the pieces of DNA that are not tightly packed away. They're open, so they can be transcribed by RNA polymerase. Whereas heterochromatin, these are very, very tightly packed, and they cannot be transcribed. The transcription is inactive. One really cool example of this is going to be calico cats. If you didn't know this before, this is interesting. All calico cats are females, and here's why. Females have 2 X chromosomes, and on the X chromosomes of these cats is going to be the allele for hair color. So you can see in this cat here, we have one of the X chromosomes, because remember they're diploid, so they have 2 copies of the X, of the sex chromosomes. In this case, it's a female, 2 X's. And one of those X sex chromosomes is coded for orange coloring, where the other is coded for black coloring. And in each, only one of those X chromosomes is active. The other one is tightly packed. It's called a bar body. It's really, really tightly packed like this heterochromatin, so it cannot be transcribed. And the other one is left as euchromatin. It can be transcribed. And it's random in every single cell which one is going to be a bar body and which one's going to be transcribed. So it leaves this kind of patchy all over the cat, which is really, really cool. Okay, and then you have something called epigenetic inheritance. This means even though 2 individuals may have the exact same allele for a gene, one of them may be turned off and one may be turned on just because of how they're packed. How tightly are they packed around histones? And you can see this in this fly here. I'm going a little quickly so that I can get through this in the 15 minutes I have. Okay, so then we also have something called post-transcriptional control. So you can see this is after transcription has happened. So we're still in the nucleus. We're looking at transcription right after it has happened. Remember we had those exons? These are the genes that are expressed, the pieces that are expressed. And we had the introns that were in the way. This is the pre-mRNA. Now, before the pre-mRNA becomes mRNA by splicing these introns out. Once those introns are spliced out, then the exons are put together, and they can be put together in different ways, and that can make different proteins. Here's protein 1, and here's protein 2, all from the same gene. So that's another way to regulate how our genes are expressed. So post-transcriptional control, changes in the actual mRNA message, and that has to do with how the introns and exons are put together. All right, eukaryotic regulation, translational control. We can see how mRNA alterations affect translation, not the actual message, but whether it's translated or not. Okay, post-translational control, how long is the protein active? How fast does it degrade? And there's a lot of factors that play a role in this. And this is going to be a review here. The 5 primary levels of control, this is what you really need to know for your quizzes and your tests. Chromatin structure controls which genes are accessible. Transcriptional control controls if mRNA message will even be made. Post-transcriptional control changes actual mRNA message. I'm going to have to stop there, and I will have another one for the rest of this chapter.